Okay, welcome everyone to the European Society of Marine Biotechnology. Today, I'm just going to quickly go through, first of all, thank you for signing up and registering to this event. This event will be recorded and we will upload it in our ESMB webpage, so you can go in and have a look on your own time. So currently, I'm just going to share my screen with a couple of slides just to give some background information to those who have not joined our society before. Okay, and I'm going to share my screen. Share. Right, so hopefully everyone can see me. Everyone can see the screen? Excellent. So, as I said, this is the European Society of Marine Biotechnology, and this was established in the 26th of April in 1995 in France. And the idea was to promote the biotechnological potential in from the marine environment across Europe, but it seems like it's across the world as well. So what ESMB promotes is exchange of scientific information, training and cooperations for its members that benefit from this, and the society as well as the environment. What kind of activities we cover? Of course, we have our every 2-year ESMB conferences. This year has been postponed for obvious reasons. And we have our webinars, which is a new addition in response to this lack of communication because of COVID-19 and so on. And we are very happy that it's been going quite well so far. The other activities include meetings and training, and of course access to the journal from Springer Marine Biotechnology and our newsletters. Another thing that is actually going to be resumed soon, and this is going to be, I'm going to repeat that at the end, is our students. There is exchange of students, and we support placements through the Erasmus Plus and other schemes. So this has been postponed and paused for a little bit for the because of the COVID-19 situation, and this will hopefully be resumed maybe September, October time. But obviously we will keep everybody informed about this. So this is our main web page. And today I'm going to be presenting Professor Jeanette. Sorry, because my screen is a bit on the way of just a second, of the full title. There you go. Jeanette Andersen, who is coming today from Norway, the Arctic University of Norway, which I found an extremely cool name for a university. And she will be discussing about sustainability, from sustainable resources to novel marine nutraceuticals for management of metabolic syndrome. And this is their newly funded SuReMetS project. So Jeanette, Professor Andersen is a professor in marine biotechnology at UIT and the head of the natural products platform in Marbio at UIT. Marbio has expertise in screening of extract libraries of natural products, including terrestrial and marine microorganisms, as well as products. Professor Andersen has successfully characterized several novel bioactive compounds, and the Marbio, the center itself, has obtained funding through the National Infrastructure Program of Norway, and it is a specialized node in the European Research Infrastructure Consortium. Professor Andersen's research interest and focus is the discovery of novel bioactive compounds from the marine environment. And today she will be talking to us about the SuReMetS project. So I hope I introduced you enough, Jeanette. So I'm going to stop at the moment sharing my screen. And I'm gonna pass the baton to you. Thank you very much.
Thank you very much, Maria, for the kind introduction, and also for the participants to endure the long title of this project, to learn more about this. So I will start sharing my screen in a second, and hopefully you can now see it. Can you see my presentation, Maria? Yeah. Okay, thank you. So the SuReMetS project, as Maria said, from sustainable resources to novel marine nutraceuticals for the management of metabolic syndrome. This is a Blue Bio co-fund project that I will present to you today to get to know this project. It's quite new as well for us. But first of all, let me click in to say metabolic syndrome, what it is and why should we bother with this. So metabolic syndrome involves a variety of conditions, including high blood sugar, high blood pressure, excess body fat, and abnormal serum cholesterol triglyceride levels. So there are many conditions involved in metabolic syndrome, and we know that this is getting a bigger and bigger problem in the world. So it's important to keep in mind of all this and try to prevent this from happening. So how do we deal with the metabolic syndrome? So of course, first line treatment is lifestyle changes for physical activity and weight reduction, and also improvement of several risk factors related to METs. And we know a lot from the literature that fish consumption is linked with many health benefits due to their high protein levels, but also to the other contents of unsaturated fatty acids, vitamins, and minerals. And also there's a lot of literature saying natural compounds and some dietary ingredients have really a positive effect already to do management and trying to prevent and treat metabolic syndrome. So, and in particular, there is a lot of literature. This is a modified table from a great review from one of our partners in the project, bioactive peptides from fish protein bioproducts. This was a very long, big table, but this, I hope this illustrates just by I took a part of this, that Atlantic salmons were byproducts, trimming, skin collagen that have been enzyme treated have given a variety of biological activity. So here it covers from antioxidant, cryoprotective, anti-inflammatory, and DPP-4 inhibitory, which is related to diabetes and glucose metabolism. So I think this, just by picking a part of this table from this literature reference to show that there is a lot of documents and literature referencing the biological activity, especially coming from fish byproducts and from underutilized fish species examples. So this inspired the project, and I will come into more detail about the project itself. But first, I just want to introduce my group, which is coordinating the SuReMetS project. So Marbio, we are at the UIT, the Arctic University. So we are a platform for identification, isolation, and characterization of novel bioactive compounds. So we are doing marine bioprospecting. So we work with marine extracts that we are doing purification, identification, and screening of. Of course, this happens in a circle, and the main objective is to identify active compounds. Since also we work a lot with marine microorganisms, we are also now looking into the genome potential, not only the chemical potential. So trying to go dig through also the genome to see if we can get access to the active compounds by selecting the most promising producers. And as Maria mentioned, we are a part of the NOR Open Screen, which is an infrastructure program in Norway. And we are an approved specialized screening sites in the EU Open Screen, which is part of the ERIC, the European Research Infrastructures. So our infrastructure is built up by setting up various automatic liquid handling systems to do the bioactivity screening. And we also have a microbiology unit where we can cultivate and work with marine microbes, having various shake incubators, but also an automated colony picker machine here to work with more high throughput testing. And of course, the chemistry unit to try to identify and purify the bioactive molecules. We have a set of various machines and chromatographic systems to isolate and identify. So we are very happy that we could utilize this marine, infrastructure to work with various projects. So my expertise is mostly on bioactivity screening, and I just took this table just trying to show the different assays that we normally work with. It covers quite broadly from anti-cancer, antimicrobial, but also immunomodulatory, skincare, diabetes, toxicity. And one of the recent additions we have is that since we have developed some projects, is the ADME, so the preclinical and like druggability parts of the molecules when we have identified this for microsomal stability, metabolic identification after degradation, and this permeability assays is something new that we just added to our list. And we see that this toolbox is developed over time, and we have new things coming in and new things going out with depending on the different projects that we are running. But this is sort of a baseline, what we normally have part of our toolbox to screen marine microorganisms or other marine organisms for bioactivity. So this is sort of course, this is the natural products discovery pipeline, like many of you know, starting with the marine organisms here. We do have activities involved in collection, testing, identifying the novel chemistry, then involved in biological characterization, target identification, and then often the chemical optimization of the compounds that you identify originally. And here on the preclinical and clinical development, of course, the industry involvement is very important. And these pictures shows what I mostly have experienced is working with marine invertebrates and microbes. So most of our projects have been more like drug discovery and focused on small secondary metabolites. And we had various projects like these that are in different phases. Digibiotics is an antibacterial project. Markin is a breast cancer project where we have active compounds, and then this TechAML project is a blood cancer project where we are now doing optimization chemically and trying, hopefully by this fall, can enter animal testing with the selected lead compounds here. We are also involved in a pain project where we are trying to also go through the preclinical phase. But most of all, we have projects covering the whole pipeline of the simplified pipeline here shown by natural products discovery. And SuReMetS, which is the topic for today, it sort of cover this part of the pipeline all the way to preclinical, but it's other resources, is underutilized fish biomass and algae that is in focus. And now I will switch over for sharing more detail about this project. As I mentioned, it's Blue Bio co-funded, and the material here, I will come back to more about which material we have selected, but it's the biomass processing of underutilized fish and microalgae biomass. But at the same time, we want to look for novel enzymes to do the biomass processing here. So we will mine microorganisms throughout this project on the same time where we are starting to process with known enzymes. And the application testing, of course, evolves around the metabolic syndrome, and we have selected diabetes, antioxidant, and anti-inflammatory as the main assays to be performed. And one of the main goal here is to get closer to a scale up, and we have industry involved. And we really also want to get started the preclinical tested, do the ingredient valorization, market evaluation, and in the end, we want to be able to communicate that we are working with natural, safe, and well-accepted products. But this is, of course, something that when we interact with people like you listening in here, is of course important to get feedback. So the whole goal here is to find novel biofunctional marine ingredients with beneficial effect on biomarkers for metabolic syndrome. So we are 7 project partners. It's us here. We have 4 research partners from university here in Norway. But we also have partners, University College of Cork, Ireland, University of Limerick is participating from Ireland, and we also have University of Hamburg as partner. And then we are very lucky to have 3 industry partners on board: Biomarine Ingredients from Ireland, Blue Biotech from Germany, and Materialis Innovation from Norway. So this is an industry-driven project where we are following and discussing with them closely, because they will be the one that are providing the raw material, but also do the valorization of the products in the end. So the main objective, as I already touched upon, but is to the first main goal is to identify enzymes that can be used to process the biomass with higher efficiency at low temperature. So this is one of the main goals. The second main goal is, of course, to identify the marine ingredients for targeting metabolic syndrome. So the project were to start last year in June, and it's a 3-year project. But I put a mark here on here because of COVID-19, some partners were allowed to start later in September and to get going with this project because there were a lot of issues with lab access and there were some delays in getting started. But now all partners are up and running and are delivering results. So this is an overview of the work packages in the project. And I will, in addition to this figure, try to walk you through the different steps. It's the biomass processing here, byproducts from fish, microalgae, and our main goal is to use enzyme-aided extraction here to get out different chemicals, lipids, peptides, metabolites before we test them for in vitro bioactivity testing, but also the chemical analysis and hopefully also some mode of action. The scale up and the ingredient valorization at the end, lower part here. So you see the enzyme discovery is sort of like a add-on on this side, but it's as important to find novel enzyme, but also to find new ingredients. So if you can do both, that would be great. But also if you could use existing enzymes to get new products, it could also be a valid goal for this project. So where are we now with the biomass process? So we have started some of the hydrolysis. It's a partner in Limerick that is doing the initial part of this. They started with 3 commercially available food grade enzymes so that we didn't have to wait for the discovery of new enzymes or microorganisms. And already we now are evaluating the degree of hydrolysis, the molecular mass distribution, and there is a selection and shortlisting of biomass species. And one of the important part of the initial of the project in the beginning is the feasibility and risk assessment of the biomass at industrial scale production, because we don't want to start with biomass that will not be able to scale up either for access of raw material or enzyme deliveries, etc. So this is an important part to assess already from the beginning. So here, it was the biomarine ingredients in Ireland leading this task. They did it together with Modalis Innovation, which is the supplier of the fish biomass, both from Norway and Ireland is the fish biomass supplied. And this is a short list per now that were risk assessed and feasibility for the scale up, etc. that went through. And per now, this is the list that we are working on. And I also added this to microalgae species with Blue Biotech in Germany is supplying. So there was a lot of desktop work taking out this, having overview of accessibility and processing limitation, etc. Because it's the biomarine ingredients that will do the scale up in the other end. So it was important to have their opinion from the start. This is a picture of the Blue Biotech company in Germany. They have already products on the market. They have this nanocorruptives biomass that they use for aquaculture application. And their goal of participating in this project is to expand the product range and see if there are other valuable molecules, if they can increase the value of these algae. So coming back to this enzyme discovery and using bacterial diversity, it's a very complex work package. So it's Cork and Hamburg and Limerick working together on this. And the main thing here is that they use prokaryotic isolates from the microalgae and fish guts, as well as their microbiomes to exploit to see if we can find in the source that we want to degrade and hydrolysate, if we can find some novel bacteria that will efficiently hydrolysate these biomasses. So they will use metagenomic strategies and use enzyme assays to try to identify the most promising strains that do have these activities. And of course, there's a lot of bioinformatics involved here to really go through the metagenomes that are being produced in the project. And of course, trying to express and access these novel genes that we can really use to see in the end if they could be used in enzyme-aided strategies to treat the microalgae and fish biomass that we have access to. So I have a slide here from University of Hamburg, just overviewing the work like in very general, but using the metagenome library, as I mentioned, from the algae and from the fish biomass that we are working on, trying to characterize and identify promising biomolecules in the end, but going through the isolates and strains to really try to find novel enzymes and processing materials. And hopefully we will have time, and that's the main goal, to implement this new enzyme in the process before the end of the project. So there's a huge activity, and I'm very glad that we have Cork and Hamburg on board of this work, and they are splitting their work between the algae and the fish biomass. So they are really digging down into the details here, finding promising candidates from the microorganisms. So this is just an initial picture. I got this from Marine at Cork. This is salmon skin that she started to isolate bacteria from so that we could see if we have here. This is a picture of an enzyme assay that she used initially just to see which one are producing. And actually we are in the process where we are going to start the hydrolysis of the fish biomass and algae samples with 3 of these new microbial proteolytic enzyme preparations. So we're very happy after a few months of being in the lab here that we could already start testing this in the lab scale to see if they are working out. So, and we also have set up the bioactivity testing of all of these hydrolysates to be produced. It's anti-diabetic, where we're using inhibition of DPP4, this enzyme, which is also related to appetite reducing and this glucose metabolism in the cells. But we also have an insulinoma cell protection assay up and running now, where we are actually testing for, the insulinoma cell is producing insulin, and if you have a type 1 diabetes, you have induced cell death by cytokines. And if we can protect these cells after exposure to cytokines, we hopefully can see something that will have a beneficial effect on type 1 diabetes. But we are also not transferring this assay to trigger cell death in this insulinoma cell by using high concentration of glucose or free fatty acids, so that we are mimicking what happens in type 2 diabetes, and then trying to see if we can protect these cells after this exposure with our fish hydrolysates or algae hydrolysates. So we have some initial data running here, but we need to do more, but at least the cytokine protection assay is up and running, and a good one for type 1, but we are still optimizing it for this glucose and FFA. For the anti-inflammatory, we're using inhibition of LPS-induced production of TNF-alpha in monocyte cells, human monocyte cells, and this assay have been running for a long time. So here it's just running through the samples as they arrive in the lab here in Tromsø. And for the antioxidant, it's the biochemical frappenorac, which we are using to measure the ferric reducing ability with this assay in the presence of antioxidants. And in the ORAC assay, which has a different readout, but it also includes the oxidant and the antioxidant at the same time. So that's an advantage of this assay. And these 2 assays has different readouts, which I will say this is an important thing if you're going to work with highly pigmented things or colored stuff when you're running the assays. When we have identified bioactive extracts, we will use our chemical platform here using UHPLC and high resolution MS-MS to go through and really characterize the extracts, what kind of peptides, carbohydrates, lipids, and if there are other metabolites. And we want to do the major classification of type of compounds, and also look at size distribution of peptides and carbohydrates, and hopefully also be able to look on lipid classes, and this is particularly interesting for the microalgae samples. So we do have some initial bioassay data. This is a master student that's been running in the lab. So this is a FRAP assay where we have done a concentration range of some of hydrolysates that were available for us at this time. And we could see a dose dependent that we got higher antioxidant activity with higher concentration in some of these initial hydrolysates. And with this DPP4 assay, which I mentioned was for this anti-diabetic or glucose metabolism. We also see that we have very high activity here on with good activity, and it's somewhat dose dependent. But what we saw here is that we do have some issues with background readout for this this was absorbance readout. So we're now transferring this for a fluorescent readout so that we can with doing this in another way. So more data is to come. But we are getting some experience working with the fish hydrolysates in comparison to other extracts that we worked a lot with previously. So it's nice to run them through different of the sets of the assays. The last part is, or one of the important part, as I mentioned, is the ingredient valorization, which of course is in the future for us here, but already now it's important that the market evaluation of what we are aiming for is done by the industry partners. So we want to detect any of the effects in the extracts in a preclinical study and really like to confirm this. And we have to already now look into what kind of animal models will we use to demonstrate efficacy, and what kind of mode of action that we will have in humans. Of course, this will depend on the bioactivity that we are picking up. So it's a little bit too early to select the animal model already. So we will have to do that case by case for the new bioactivity that we do detect in the disk. But it's going to be, I think, based on the initial data we have, it's going to be a tough choice, because I think we're going to have many candidates extracts to choose from. So I'm really looking forward to this. But this is run and led by Madalis, which is one of the industry partner that will make the decision based on the results that we obtain in the project. So we have discussed a lot about this. One of the things in the Blue Bio is like the technology readiness level of the projects, and this call was aiming from 2 to 7. So number 1 is like basic principles, 2 is concept that is formulated and you're moving it through proof of concept, validated in lab, validated in relevant environment, and demonstrated in relevant environment. But also this prototype demonstration should be in like industry environment. So we realized quite quickly that having different biomass, the microalgae, it will be challenging to reach all the way up to 7. So we were quite early saying that for us with the microalgae, I think this is what we're at technology readiness level 5 is where we can be with the material and the technology that we have in hand. But for the fish biomass, with the biomarine ingredients, the partner which having experience in scaling up, they are quite confident that we could reach number 7 for the fish biomass. And this is quite good because working from the university, we are often very struggling around with the proof of concept and in the lab. So it's really nice to have this focus that we are quite early in seeing where we are going and what we are aiming at, and that it's not only the bioactivity itself that it's deciding, it's also other characteristics and features of the biomass that is deciding how far we can get with this. So I want to say, I think SuReMetS have a lot of opportunities. I think looking at our pipeline here, I see that there are a lot of projects working on protein hydrolysates and bioactive peptides as functional food ingredients, but still it looks like that for every case, you have to develop this to really be economically feasible, and you have to by case think about the consumers and the industry, how you do this. So it's like for every product that you are thinking about, you should really think about all the steps from the processing all the way to the market and then the ingredient valorization. And I think there's a huge opportunity for this to happen because we see the regulation for processing waste and also the abundance of underutilized species in the fisheries. So I think this is getting more attention from many, and there are many interesting projects ongoing. So I think this should stimulate like the fish processing industry to really take part of this conversion, like how can we make this low value material into more valuable products. So there is definitely a really good opportunity, and I hope that SuReMetS will contribute to build under this case. But We know that there are many projects, but we also see that there are challenges, but it is an opportunity. So of course, in SuReMetS, we have a lot of people involved. So from UIT, we have Yannick Schneider, which is a chemist. He did his PhD in marine bioprospecting. My colleague Espen is doing the chemical identification, and this is the master student Njål. In Cork, we have Maureen that's working in the lab, and the PI there is Alan Dobson. At the University of Limerick, Neda Khatib is working in the lab with the hydrolysis part, together with Padraig and Harnedy, but it's Dick Fitzgerald, which is the PI at University of Limerick. At Biomarine Ingredients, it's Nehal Geit and Jason Woolley from this industry. From Blue Biotech, it's Sebastian Lippenmeyer. And in the University of Hamburg, it's Ines Kroon and Yasha McDonnell, which is a PhD student, and the PI is Wolfgang Streit. And at Modalis Innovation, we have Jardan Røo that is participating in the project with his input. So just to end this, so why should we think about and involve ourselves in SuReMetS? So I definitely think that this could contribute to the health aspect, but not only the health aspect, it's also the bioeconomy. So I think by digging into these resources, we can have higher value products. It could be enzymes, it could be proteins, but also other metabolites. That's sort of the factor X in this that we will also look into. So I think that these fish byproducts and algae, together with all the technology that are being developed, can contribute with sustainable solutions for the future, and also answer on this blue bio call is to really take advantage of underutilized species and really create contribute to the bioeconomy. So I would just thank you there for your attention. And if you want to follow on LinkedIn, we have the SuReMetS project, a page with Yannick established. Our research group is also on Instagram. And I can also mention that the European Society of Marine Biotechnology is also on Instagram. And if you want to, you can send me a message, either an email, but also on Twitter. And we are very grateful for the Blue Bio Co-Fund to fund this project. Thank you. So
Thank you very much, Jeanette. That's an excellent talk. And also I would like to highlight that you're a board member of the European Society of Marine Biotechnology that I forgot to mention earlier on. And yes, that is an extremely interesting project. Biotechnology and bioprospecting is always something of great interest. I think at this point, I would like to invite everyone, if they want to ask something, either by through the chat, or if they want to unmute themselves, like you can have the function of raise hand, and then you can just unmute and pose your question. Or if you have something on the chat, then I can read it for you and ask Jeanette. So does anyone have any questions at this point? I think I'm going to start actually, because oh, that's very exciting. It includes microbes, it includes so many byproducts, and it's extremely interesting. So you will, I saw that you will be isolating Hamburg, you said, university, they will be looking at the microbial world and isolating from the byproducts microbes. And you had some initial results from the salmon skin.
Yes.
What was it for protein hydrolysate, that one?
Yeah, so that was a proteolytic activity in those, yes.
And was it from the top of the skin? Do you know which area of the skin?
So this was done in Cork, so I have to think about, I will not be able to answer accurately. I could do the check, but they have been working with both the fish guts and I have to have a small thinking. It might be from the fish gut that she actually has the strain, but she started also working on the skin. So there was all the details, since this is commercial also interest in this project, a lot of details on the strains, etc. I couldn't share at this moment, but hopefully during the project we could be more open about some of the data, but this is very initial data that we started, but I'm really happy that we just able to actually find some new enzyme activity.
Great, that is something I would like to follow up. Always anything that any microbe that grows on other organisms, always of interest, isn't it? I have Sergey, who is nodding along. Dr. Sergey Dobrechov, who is the president of the European Society for Marine Biotechnology and has joined us, and he has a couple of questions. Hello, Sergey. Would you like to say your questions, or do you want me to read? Okay.
I can read it. He's asking about drugs against COVID-19. That's interesting. Actually, I have had a lot of requests since we do have a screening facility here that if we could do some screening against the viruses. So we have a lab that has done herpes simplex virus screening. And if I get more project requests about COVID-19, this might be actual, but part of the EU Open Screen, they are running now COVID-19 with the chemical library of EU Open Screen. It's involved in COVID-19 projects actually. And I guess you all heard about this plitidepsin from PharmaMar in Spain. That's nothing to do with us, but one of these marine compounds that were in development for cancer, actually, they quickly moved it into, I think, phase 3 trials against COVID-19, and it looks like it already have quite good effect because they could rely on safety data from the cancer trials, and it had really good effect on this. So hopefully it would be really interesting to participate. But what we will do is that if we have some of our metabolites and compounds, we will include that in the European library. And hopefully then it will be screened by others for COVID-19 in the future. But for now, we are not doing it that in-house here. You're saying about blood clogging and inflammation. So we are running anti-inflammatory assays and blood clotting, etc. Not focused, but we have very interleukin cytokines that we could measure, and we have interleukin 1 beta 6 that we are monitoring, so that's something that could be like indirectly involved in these compounds. So we hopefully, when we screen our hydrolysates, we will map their anti-inflammatory effect, and maybe if it's really interesting, mode of actions, etc. And there are a lot of pathways that we then could interrogate with this following further. So I'm looking forward. I hope we'll be in that position after these years that we could dig into inflammation even further, but it's a very complicated field.
Yes, I can imagine. Any other questions? I think it's a midweek. It's a little bit difficult, especially now with exams being started. Well, getting there, being prepared. So I have one thing also to follow up. Well, actually, no, I do have one more question. You did mention that upscaling is one of the most kind of difficult parts. What are the main issues with upscaling in general?
Well, first of all, you could say just the supply of the biomass, so that if you could talk about tons, etc. So of course then, but for some of the fish species like biomarine said, that's not a problem. They have enormous amount. But then again, if you're going to do the enzyme, we have to make sure that the enzymes are available and working properly in large scale. So I think it's the cost of producing these enzymes as well is also could be a limiting factor, I think. So of course, it's not sufficient to have a good enzyme, but if it's super expensive to actually get sufficient amount in this large scale. So I think there are many aspects. It could be everything from the supply, it could be the process itself and how to mix it properly. But this is something that I just recently discussed with the biomarine ingredients in particular, and I realized there are so many things. So, and it also has to be adjusted to the industry side so that it shouldn't be difficult for them to deliver the waste material if that's sort of introducing a costly step. So it has to be all this like mechanical, all these tanks, etc. There are so many things to consider. So I'm not the expert on that. So, but hopefully I will learn a lot. But I think there are many things. So I just listed a few of the things that I would expect, and I heard from the discussion that could be a main thing.
That's a very interesting. I didn't even think of a couple of ones that you just mentioned. Very interesting to see that from that point of view as well. Very interesting. Yeah, I think I don't know if Sergey or anyone else has another question. At this point, okay, Katerina. Hi, Katerina. Nice to see you here. Would you like to tell us what's your
Hello,
question?
hello. I'm gonna show my face. Thank you for the interesting talk. I must say this might be a stupid question, and I must admit that I unfortunately missed the first part of the video where this might be addressed. But you were talking about metabolic syndrome and trying to find enzymes in marine microbes. I was wondering, because there's quite an extensive research into the human microbiome and enzymes produced by our gut microbiome. How the marine enzymes, the enzymes from the marine microbiomes are more efficient, or how are they superior to the ones that we should be harboring in our gut? I'm sorry if this is not relevant.
I don't think it's a stupid question. It's definitely a valid question. So of course we could look in the human microbiome. So one of the things that we think about the marine ones is that they are perhaps more cold adapted, more efficient at lower temperatures, because they are in our body it's 37 degrees and most enzymes are perhaps optimized for that temperature. But also the thing is that we go to the source to look for microbes. So like if it's from a salmon that we want to chew, it's in that environment we would look for microbes that might be able to degrade it efficiently. So because they have co-evolved, so we do expect that in the surrounding of these marine organisms that they will have microbes in their environment that are efficiently in using there as a resource, as a nutrient source, so that they will be chewing them in a more effective way. That's at least what we would expect. But also the temperature is of importance here.
Thank you very much.
Yes, thank you very much, Katerina, for your question. Yeah, it is the temperature adjusting the temperature for enzymes to work is, I think, a big challenge, also for processes, industrial processes, but also for different applications. I'm aware. So I think at this point, I have one small announcement that at the moment we are organizing a little mini European Society of Marine Biotechnology symposium, which hopefully will take place a couple of, I think, mid-June at the moment. And it's going to be of postgraduate students that they would be, it will last half a day, I believe, and then just small talks on their projects. And very, very exciting, because looking at early career researchers and postgraduate students it's at the heart of the European Society of Marine Biotechnology. So I will be sending information out on this hopefully soon, once we have the final line of who is going to be talking. And with that note, I think I don't have anything else to add. I'm just looking forward for the next mini symposium. And I will be, of course, do join us on Instagram. Sorry, I forgot to put the actual exact Instagram address, but if you put ESMB on Instagram and LinkedIn and I think and Twitter, I believe, you can definitely find us and follow us. And of course, on the European Society of Marine Biotechnology website, esmb.org, where we upload hopefully in the next one day or two this recording as well, so you can go and visit and watch it at your own pace. So with that, I would like to thank again Professor Andersen and everyone for joining, and I will hopefully see you in our next ESMB series. I'm stopping recording now.